Hi, I'm Sarah Al-Noubani. I'm a chemistry major. My project has been over the biochemical investigations of F420-dependent glucose-6-phosphate dehydrogenase from Cryptosporidium arbor. F420-dependent glucose-6-phosphate dehydrogenases, also known as FGDs, catalyze the oxidation of glucose-6-phosphate using the cofactor F420 as a hydride acceptor, generating 6-phosphogluconolactone and the reduced cofactor F420H2 within the pentose phosphate pathway. This has previously been studied in FGD from Mycobacterium tuberculosis, termed MTB-FGD, due to its medical relevance in treating multi-drug resistant and extremely drug resistant forms of tuberculosis. It's involved because the reduced F420 cofactor is implicated in prodrug activation against these highly resistant forms of tuberculosis. Currently, FGD is being studied in a less well understood gram-positive soil-dwelling bacteria, Cryptosporidium arvum. FGD from Cryptosporidium arvum, termed crier FGD, exhibits broader substrate specificity toward multiple sugar phosphates, such as glucose-6-phosphate, fructose-6-phosphate, and mannose-6-phosphate. This substrate promiscuity is the main reason why this enzyme is being studied, as improving our understanding of FGDs can expand our knowledge on this cofactor and this enzyme class, which can reveal more about treating tuberculosis. The F420 cofactor is made up of an isoalloxazine core, where upon the addition of a ribitol motif, this becomes the precursor FO. Upon phosphorylation, this becomes F+, and upon the addition of a lactyl group, it becomes F420-0. Depending on the organism, 0 to 12 glutamine repeats can also be added to create a polyglutamyl tail. F420 is spectrophotometrically distinct in that upon reduction to F420H2, it loses its maximum absorbance of 420 nanometers and instead gains a shoulder at around 300 nanometers. As a result, a UV spectrometer can monitor changes in absorbance over time and be used to determine the reaction rate and be used for kinetic characterization. The kinetic experiment I will be primarily focusing on is the steady-state experiment. In addition to Crier's dehydrogenase activity, previous NMR studies have shown for it to have isomerase activity as well. This is the crystal structure of MTB-FGD. It is a 78 kilodalton homodimer, which means it's made up of 2 identical subunits. This is a closer look at the active site of this protein. The amino acids of Crier that have been identified as analogous to those in MTB-FGD's active site have been found to be histidine 38, tryptophan 42, and glutamate 107. Crier FGD has not been structurally characterized, so we cannot be certain of its active site unless catalytically characterized. Conducting the temperature dependence and pH profile will reveal what amino acid is functioning in the active site, and inactivation experiments will serve to confirm this. To conduct any of these experiments, firstly the enzyme must be obtained. The FGDs from Cryptosporidium arbum are expressed in an E. coli host cell where they combine to create the transformed cell. Then these transformed cells are grown on an agar plate, and from this, a colony of the agar plate is selected and grown in terrific broth under these conditions. Once they have grown, induced, and expressed, they are harvested and purified using fast protein liquid chromatography. SCS page gel electrophoresis is used to confirm overexpression and purity in combined fractions. In this gel, this was successfully achieved in fractions 61 through 70. This confirms that we're able to successfully purify our protein and can move forward with the kinetic experiments. The temperature-dependent studies were conducted in a steady-state format under these conditions, where the catalytic efficiency, also known as kcat, of each pH and temperature condition was obtained. In terms of temperature conditions, temperatures of 25 to 40 degrees Celsius in 5-degree increments were tested, and in terms of pH conditions, pHs of 6 to 9 were tested in 0.5 increments. Plotting the logarithm of kcat versus pH for each group temperature yields a bell-shaped curve. This bell-shaped curve tells us that there's an acid-base catalysis occurring, and the parameter K1 will be extracted from the fitted data. K1 will be the acid dissociation constant of whatever is acting as the active site base. Now the linearized Van't Hoff equation can be used by plotting the extracted pK1 of each temperature graph versus its inverse temperature. The slope of this graph can then be used to identify the delta H of ionization and was found to be 6.3. And in comparison with reported pKa and delta H of ionization values for functional groups, this group was assigned as a cationic imidazole, essentially a histidine. By treating our enzyme with diethyl pyrocarbonate, or DEPC, it selectively modifies deprotonated histidine residues. If upon treatment with this DEPC our enzyme loses activity, then we can confirm that there is a histidine residue involved in catalysis. The enzyme was incubated with different concentrations of DEPC, and every 30 minutes for 2 hours, an aliquot was taken and the activity of it was measured under saturating G6P and F420 conditions. For each concentration of DEPC, we fit it to the following equation, where VI over V naught is a ratio of reaction rates of the enzyme treated and untreated with DEPC. The data shows that upon treatment with DEPC, the activity of the enzyme decreases. Additionally, the fitted lines meeting at one point shows that the data is monophasic, and thus there's only one histidine in the active site that's being modified. Extracting the parameters of this plot yields an inactivation efficiency of 25. In conclusion, there is one deprotonated histidine residue in the active site that is involved in catalysis. Based on the analogous amino acids that were compared between MTB and Crier FGD, this is predicted to be histidine 38. There are various ways in which this can be further confirmed with future experiments, such as site-directed mutagenesis performed on this histidine, or structural analysis with cross-linking or crystallography.